Hey guys, this is Unit 2, Lesson 1. This is what we'll be actually going over in class. We'll be working on some things in class from this unit next class. This will cover Chapter 4, 4.1 through 4.4. You should have already done the Dynamic Study module with the summer assignment, and you should have read the sections by now. All right, so let's talk about cell structure and function. Again, we know that structure dictates function. That is a running theme throughout AP Biology. So make sure that not only do you know that, but you understand how the structure of something is going to determine how it works. And if you change that structure, you are also going to change the function. Now, so again, this is Concept 1, Day 1. Sections 4.1 through 4.4. So before we can even talk about the cell, we have to look over how we even know cells exist. And that comes from the discovery of Robert Hooke. Robert Hooke was observing cork cells, and he coined the term cell when he was looking at the cork cells through the microscope. Now, what he was actually looking at was the cell walls left from plant cells, but to him, it looked like the rooms that monks slept in, so he started calling them cells. From there, Antoine von Leeuwenhoek was able to make a better microscope, and he was the person to first observe living cells in droplets of pond water. And so it's important that we know how microscopes work and what's the purpose of microscopes, because without a microscope, we would not be able to even observe cells, or even now we can observe things even smaller than cells like those organelles. Now there's three parameters that are extremely important when it comes to microscopy. The first one being magnification, and we know magnification is how big or how small something appears to be. You know, when you magnify something, you want to make it bigger. So I am going to pull up a picture on my phone of my beautiful children, my chitterlings. All right, so magnifying this is how big or small. See, I'm magnifying it when I make it bigger. This is magnification. Now, another thing that you're going to notice, another parameter of microscopy, is the resolution. If you could see, which you probably can't, but the bigger I make this, the more grainy it looks. So that means I am distorting the resolution, okay? The smaller it is, the better the resolution. And another parameter is going to be contrast. And contrast is just how good can I see the difference between the objects I want to see and the stuff that's in the background. Now, another thing I want you to observe about magnification is the field of view. I want you to notice that the bigger the magnification, the smaller the field of view. So if I want to see everything, I need to decrease my magnification. But if I just want to see more details, I'm going to increase the magnification, but that's also going to decrease my field of view. Okay? So, three parameters of microscopy: magnification, the ratio of an object's image size to its actual size; resolution, which is the measure of clarity of an image; and contrast, which are the visible differences in parts of the sample. Now, in here, we're going to use a light microscope, and in most labs, you will use a light microscope or LM. A light microscope basically uses a system of lights or mirrors, and the visible light is going to pass through a specimen, then it passes through lenses, and the lenses are able to refract, which needs to be in that image so that it's magnified. And so when you have a light microscope, this is like the one we have in the lab. We have two sets of lenses that actually magnify. You're going to have your ocular lens, which is the one that you actually look through, and I think our microscopes have two. We may have one. No, we have one. We have a single ocular lens. And then you have your objective lenses. And so the way you find your total magnification is you multiply your objective times your ocular. Your ocular is going to be 10 magnification, and your objectives can vary anywhere from 10 to, I think the ones we have go up to 400, but I think they have some microscopes that go even higher, probably up to 1,000. There's various techniques that can be used to enhance the contrast or the difference between what you want to see in the background and using a light microscope. And one of the best things about a light microscope is that you can actually use it on living things and you can see color. That is a big difference between the electron microscope, which is what we'll talk about later. Now, with the microscopes that we have in most light microscopes, the smallest thing you can probably see is going to be like the nucleus, because most of the subcellular things like organelles and stuff like that will definitely be too small to see with a light microscope. There have been some advances in light microscopy where you're able to label molecules or structures with fluorescent markers to make them fluoresce so you can actually see them better. Confocal and other types of microscopy have sharpened the images of tissues and cells. And then there are actually new techniques and more powerful light microscopes that allow you to see some structures as small as 10 to 20 micrometers. But again, we don't have that kind in here, so yay. Now, let's talk about the electron microscope. There's two types of electron microscopes. You have a SEM and a TEM, SEM or scanning electron microscope, and TEM, your transmission electron microscope. And a scanning electron microscope is going to do just that. It scans the outer surface of your specimen so that you can actually see the structures on the outside, that's those surface structures. It produces images that are going to look three-dimensional. So whenever you see those three-dimensional images from an electron microscope, that's a scanning electron microscope. Your transmission electron microscope is actually going to focus a beam of electrons through the specimen so that you can actually see the internal structures of that cell. Now, one of the things about electron microscopes is that it completely destroys your sample. So you cannot see motion. You cannot have a living sample, and you cannot reuse the sample. These are all things that you can do with the light microscope. Benefits of electron microscope is you can see smaller things, and it can produce 3D images for you. But again, it completely destroys the sample, and they're pretty expensive. So we definitely don't have one of those here at the school. So when we think about a cell, we often think about something that is so microscopic that you can only see it with a microscope. But think about a chicken egg. That's a cell, or a frog's egg. So there are certain things that you're able to see with an unaided eye. But the light microscope is going to go in the range from about frog egg down to probably bacteria if you have a super resolution light microscope. Electron microscopes are going to be able to allow you to see most of your organelles and things inside of plant cells, all the way down to you can actually see proteins and lipids. So again, you can see things that are extremely small with electron microscopes that you cannot see with light microscopes. The light microscopes that we have in lab, we may get down to 10 micrometers, so you may be able to see the nucleus, but you will not be able to see like in detail bacteria and mitochondria and things like that. Now we can actually see some bacteria when we do a gram stain, which we should be doing later on this year. So again, in talking about the cells, key things to remember about a cell: the cell is the fundamental unit of life. It is the basic unit of structure and function of living things. If something is alive, it is made of cells. That is one of the key distinguishers between living and non-living things. If it's made of cells, it is living. If it is not made of cells, it is considered non-living. It's the simplest collection of matter that could be considered alive. All cells are related by their descent from earlier cells, which means you get cells from cells. Cells don't just materialize out of thin air. And cells can differ substantially from each other, even though they share common features, okay? Two main classes of cells: your prokaryotic cells and your eukaryotic cells. The prefix pro means before. Karyote means nucleus. So prokaryotic literally means before the nucleus. There's two domains of prokaryotes, archaea and bacteria. And each one of those domains has its own kingdom. The archaea domain has the archaeobacteria, which is roughly translates into ancient bacteria. These are going to be your extremophiles. These are going to be things that live in like hot sulfur springs, extremely hot temperatures or extremely cold temperatures. The bacteria that we're more familiar with are from the bacteria domain, and we call those eubacteria because the prefix eu, EU, means true. So we consider those our true bacteria. Now, a couple things to remember about prokaryotes is that they are bacteria. If it's prokaryotic, it is bacterial. If it's bacterium, it is prokaryotic. Now, the difference between prokaryotes and eukaryotes, the major difference, the biggest difference that is most noticed is the lack of a nucleus, because again, prokaryote means before the nucleus. These organisms have genetic material, but the material is not separated from the cytoplasm. The area inside of the cell where the DNA is located is called a nucleoid region. Prokaryotes will have a plasma membrane. They will have ribosomes. They will have genetic material, and they will have cytoplasm. Some of them actually have structures such as a capsule or a flagellum, which is like a tail for motion, or cilia, which is like little hairs, and also they may have a pilus, which is considered a reproductive cilia. The DNA found in prokaryotes is going to be a circular chromosome, unlike our linear chromosomes. So the DNA in a prokaryote is going to be circular. Can you see this? I can't draw a circle. But what we're used to seeing are linear chromosomes in our types of cells, in eukaryotic cells. So that is a huge difference between prokaryotes and eukaryotes, that circular chromosome. There's no membrane-bound organelles found inside of prokaryotic cells. Now, the way I describe a membrane-bound organelle is if you were to imagine Jell-O with fruit in it, okay? The Jell-O will be the cytoplasm, the fruit would be the organelles. Well, in a prokaryotic cell, any organelle that is found in that cell is going to be directly in the Jell-O. So that's why you don't have that many. You have the ribosomes. But in eukaryotic cells, each one of those organelles is like wrapped in aluminum foil or something and then put in the Jell-O, because the job that's happening inside of that organelle could damage the rest of the cell if it was not protected or isolated from the cytoplasm. The cytoplasm is the site of most metabolic activities. And so in eukaryotic cells, the different jobs are compartmentalized into those organelles, and you want to keep that separate from other jobs. So in order to do that, you wrap your fruit, or the organelles are wrapped in a plasma, another type of membrane. And membranes are made of phospholipids. We'll get to that later. All right, so prokaryotic cells are also noticeably smaller than eukaryotic cells. The largest prokaryote is about the size as the smallest eukaryote. Speaking of eukaryotes, again, the prefix eu means true. Karyote means nucleus. So eukaryotic cells have a true nucleus. There's only one domain. That domain is eukarya. And in that domain, there's 4 kingdoms. And I say roughly 4 because there's still some argument on whether there's more than 4 kingdoms. Being that you have the kingdom Animalia, which are your animals, Plantae, which are your plants, and fungi, which is fungus. Well, then there's that kingdom Protista. Protista is what we call the junk drawer of the eukaryotes, because things in that kingdom are there because they didn't fit anywhere else. And currently the Protista kingdom is being divided into other kingdoms. So, but we're going to go with 4 kingdoms. So protists are things that are animal-like, but not animals, plant-like, but not plants, or fungus-like, but not fungus. So it's kind of like the junk drawer. The only thing protists have in common with each other is that they are eukaryotic cells. The chromosomes found in eukaryotes are going to be linear. They're going to be enclosed, separated from the cytoplasm in a nucleus, surrounded by a nuclear membrane, which is a double membrane that contains nuclear pores to allow things to get in and out. There's going to be many membrane-bound organelles in the cytoplasm. So Golgi, ER, mitochondria, lysosomes, those are all membrane-bound organelles. Again, the only non-membrane-bound organelle found in both prokaryotes and eukaryotes is going to be your ribosomes. Ribosomes do not contain a membrane. On average, your eukaryotes are going to be larger than your prokaryotes. Again, the smallest eukaryote is about the size as the largest prokaryote. So again, here's a chart that kind of compares the major characteristics of prokaryotes and eukaryotes. Make sure you pay attention to the yeses and the nos. The nucleus is not present in a prokaryote, although there is a plasma membrane and ribosomes. There's no membrane-bound organelles in prokaryotes, and the chromosome, the DNA, is circular. Whereas in eukaryotic cells, you're going to have nucleus, plasma membrane, ribosomes, and membrane-bound organelles. And the DNA, which is separated from the cytoplasm by a nuclear membrane, is going to be linear. All right, so cells are extremely efficient, okay? And we know that the surface area to volume ratio plays a major role in the efficiency of cells. So you have this cell. I don't know what is help. Nope, kind of, sort of. Get over here. So you have a cell, right? And all of the stuff that the cell does is going on inside of this cell. Like, it does all of its processes and everything. In order to do that, nutrients have to get in. Energy is going to go in and out of the cell. So you'll have energy going in, thermal energy in and out of the cell. And then also, as it's doing its job, it's going to create waste. So those waste products have to get outside of the cell. So all of these things are going on inside of the cell. And then you have this plasma membrane, which regulates the traffic of what gets in and what gets out. So if the cell gets too big, if the volume of the cell increases too much, then the cell will not be able to perform its functions, because the volume will increase to a point where there's too much stuff that needs to get in and out to support the cell than the membrane can control. So we know that the surface area to volume ratio is important, because as a cell gets larger, it becomes less efficient. So larger animals don't have bigger cells, they have more cells. Now, for example, if you have a particular cube where it has a surface area of 54 and a volume of 27, then your surface area to volume ratio would be 2 to 1. You want to always make sure that your volume is 1, okay? So in order to do that, I'm going to divide 27 by 27, which gives me 1, and then I'll divide my surface area by 27, which will give me 2. So what that means is that for every one unit of volume, there should be 2 units of surface area. Now organisms, increase their surface area to volume ratio in several ways. Like, plants have root hairs that increase the surface area. Your intestines have microvilli that actually increase the surface area. Or you have in the mitochondria, that inner membrane is folded so that it fits more membrane inside of that volume of that cell. These things increase the surface area to volume ratio, because if I have more surface area in that same volume, that means my cell can function more efficiently. So let's look at an example, and we can see why bigger is not necessarily better. So here we have 3 blocks, okay? So if we look at the surface area to volume ratio of each one of these, so what we want to do is look at the height times the width times the number of sides, because these are cubes. So on my first block, I have 1 times 1 times 6, which means that I'm going to have a surface area of 6. On my second block, I have 5 times 5 times 6, which is going to give me a surface area of 150. But look at what I did look at what is done with the third block. Now, if you look at the volume of the third block, it is taking up the same volume as the larger block, but it's doing so by having a bunch of small blocks instead of one large block. By doing that, you increase the surface area, but you have the same volume. So if we look at the surface area to volume ratio, and these are the numbers that go along with that previous one. So if you look at, this is your first block, right? So you have a surface area of 6 for the first block. For the second block, which is the one big block, you have a surface area of 150. But for that same size block, which is a bunch of little blocks stacked together, you have a surface area of 750. Now with the one small block, the volume is 1. With the one big block, the volume, which is height times width, so that's 5 times 5 times 5, would be 125. But look here, we have the same volume with the stacks of little blocks. So when you divide surface area by volume, I have a larger numerator when I have a bunch of little things as opposed to one big thing. So my surface area to volume ratio remains at 6. The larger the surface area to volume ratio, the more efficient the cell. So remember that, the larger the surface area to volume ratio, the more efficient the cell. So let's practice this one, right? So you have a simple cuboidal, so it's a cube. Epithelial cells line the duct of certain human exocrine glands. Various materials are transported into or out of the cell by diffusion. What would the volume of a 20 micrometer simple cuboidal epithelial cell be in micrometers cubed? Now that is important that you see this cube because that means you're going to be doing length times width times height. That's 3 sides, right? So we're going to calculate the volume of the cube, which is going to be S3. S represents the sides. So I'm going to take 20 micrometers and cube it. So that's 20 times 20 times 20. And what you find out when you do that, I'll give you a second. Yay! Can you see this? 20 cubed is 8,000. All right, so 8,000 micrometers is the volume of this particular simple cuboidal cell. Now, how many 10 micrometer simple cuboidal cells could I fit in that 120 micrometer cell? So what would happen if I was to take that cell and then, take that cell and put smaller cells in it? What would happen? So if you were to take a 10 micrometer cell and cube that, go ahead and do it. Do, 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 do. Gee. All right. So if you take 10 and cube it, you're going to end up with a volume of 1,000. So how many of these 10 micrometer simple cuboidal cells can I fit into that 120 micrometer cell? Well, if you take 8,000 divided by 1,000, that tells you can put 8 of those cells. So by putting 8 cells in the same volume, you have just increased the surface area to volume ratio. So what's going to be more efficient, that 120 micrometer cell or 8 10 micrometer cells? I'm sorry, I like this stuff. I get excited. All right, so we're going to work some more of these problems in class, so be ready. All right. Practice this. I'm going to have it printed out for you when you come to class, but I want you to practice, like get you a little ruler or something and just do it at home, and then we'll talk about it when you get in class on the next day, whatever the day is. Yay. And now let's talk about some cell organelles. There's some things in the cell, they are called organelles. All right, so eukaryotic cells have internal membranes that compartmentalize their functions, right? So remember I talked earlier about how you have the membrane-bound organelles, and it's like taking the fruit and wrapping it up so that whatever job the fruit is doing, even though it's in the cell, it's not just like doing its business in the mix of everything else. So it protects the cell as well as it does what's called compartmentalization. So those are internal membranes that divide the cell into compartments, and we call those compartments organelles. The plasma membrane and organelle membranes participate directly in the cell's metabolism. And metabolism is, most people think metabolism is breaking down stuff, but metabolic processes are both anabolic and catabolic, which means it both builds and it breaks down. So those processes are going to involve synthesis and it's going to involve degradation. Now, the plasma membrane forms the boundary for a cell, and it is selectively permeable. So the plasma membrane is permeable to things that are nonpolar, small, and it is and things that are large or carry a charge, large or in charge. So that means that they're either polar or ionic, require help to get across that plasma membrane via transport protein. So that's what we mean by selectively permeable. Remember that the plasma membrane is made up of phospholipids, where it has a hydrophobic tail and a hydrophilic head. The phosphate part is PO43 negative, so it's negative, and water is polar. They both carry charges, so they love each other. But your fatty acid chains of that phospholipid are mostly hydrocarbons, mostly non-polar bonds. They do not like water. They are hydrophobic. So your plasma membrane is going to be a double membrane where you're going to have the heads. This is a head. This represents a head. To the outside in the aqueous areas of the cell. So the inside cytoplasm and the outside extracellular fluid. And then the tails are going to be protected from those aqueous areas on the inside. So that's why I say double membrane bound organelle. Now, the eukaryotic cell genetic instructions are housed in the nucleus and carried out by the ribosomes. Central dogma theory of molecular biology: DNA is transcribed to RNA, RNA is translated to proteins. All cells need genetic material and ribosomes, because even though the genetic material has the instructions can only be carried out by a ribosome. This is why both eukaryotic and prokaryotic cells have ribosomes. Now, most of the cell's DNA is going to be found in the nucleus, but mitochondria and chloroplasts have their own DNA. That's why we can't say all. So in organisms that have mitochondria, they're going to have additional DNA in the mitochondria, which coincidentally is the maternal DNA. And organisms that have chloroplasts, i.e. I can't think of the name of it. Crap. It's the algae. That's it. Some green algae and plants have chloroplasts, and chloroplast also has its own DNA. DNA, again, is a template to make messenger RNA, which then contains the code to produce a protein. Ribosomes are made up of protein and ribosomal RNA, and the translation from messenger RNA template to amino acid sequence of the proteins happens with the help of transfer RNA. Nucleus is the most noticeable organelle in the cell. The nucleus is the largest in relative size to everything else in the cell. It is surrounded by a nuclear envelope. The nuclear envelope is a double membrane that has pores that allow things to go in and out of that nucleus. The DNA cannot leave the nucleus when the cell is performing its activities. The only time DNA in a eukaryotic cell is not isolated in the nucleus is when the cell is actively dividing and the nuclear membrane disintegrates. But at that time, the cell is dividing and not performing any metabolic activities. The DNA inside of the nucleus in its relaxed form is called chromatin. Chromatin is a complex in eukaryotic cells only. It's a complex of DNA and proteins. Those proteins are called histones. And those proteins are important because they're like the spool for the thread. The DNA was the thread, and the protein was the spool. When DNA gets ready to when the cell gets ready to divide, that DNA is going to wrap around that spool so that it condenses into what we see and called a chromosome. Inside of the nucleus is an area that's a slight bit darker. It's called a nucleolus, and that nucleolus is where your ribosomal RNA is going to be synthesized, and the complexes that form the ribosome are going to be made there. Ribosomes, again, are protein factories. They are composed of ribosomal RNA and protein. It's a site of protein synthesis. Ribosomes can be found in 3 locations in the cell. They can be found free in the cytoplasm, and the proteins that they make are typically going to stay in the cell. They can be found attached to the endoplasmic reticulum, which makes the ER rough. And typically the proteins made there are going to be exported out of the cell, or they can be found inside of your mitochondria and chloroplast, where they're going to make the proteins needed for mitochondria and chloroplast. And the ribosomes that are found inside of mitochondria and chloroplast are also a different size than the ribosomes that you find in the cytoplasm or attached to the ER. The last part of this lesson is talking about the endomembrane system. Now, the endomembrane system is going to be a system of membranes. So you're going to have the phospholipids and embedded, mostly phospholipids with maybe embedded proteins or carbohydrates. But that endomembrane system are the components that are actually going to be continuous with the nuclear membrane to the plasma membrane and connected through transfer vesicles. So your components of the endomembrane system are going to be your nuclear envelope, your endoplasmic reticulum, which is continuous with the nuclear envelope, your Golgi apparatus, the plasma membrane, and then all of the lysosomes and vacuoles or the vesicles that act as transport vesicles between the ER and the Golgi and the plasma membrane, and just also just in the cell to like lysosomes clean up the cell and stuff like that. Your endomembrane system is going to be about half of the dry mass of a cell. There's two types of ER. You're going to have your rough ER and your smooth ER. Your smooth ER job is to synthesize lipids, the metabolism of carbohydrates, to detoxify drugs and poisons, and storage of calcium ions. So you're going to see a lot of your ER in places like your liver, because that's where you get, detoxification of drugs and poisons. Your rough ER is going to have attached ribosomes to it, and its job is typically to synthesize proteins that are going to be secreted by the cell. So you would see more rough ER in areas like where you have saliva glands or oral glands and things like that, where the stuff is going to be secreted from the cell. Proteins synthesized on the ER are usually transported to the Golgi apparatus. The Golgi apparatus has two sides. It has a cis face and a trans face. Cis means same side, trans means opposite sides. So the side of the Golgi that is on the same side as the ER is the cis. That's the receiving side. Proteins come in through that side. They're modified or packaged or both. And then they leave in transport vesicles from the trans face, or the opposite side, the side that is opposite of the endoplasmic reticulum. Lysosomes, vacuoles, central vacuoles, which are found in plants, are also a part of the endomembrane system. There's different types of vacuoles. You have contractile vacuoles, which in protists help to maintain the water balance and things like paramecium. And you have food vacuoles, such as those that protists use for phagocytosis, when they actually take stuff in and engulf it. Lysosomes are membrane-bound sacs of hydrolytic enzymes. Those enzymes break down things like it digests like worn out organelles or bacteria or different things like that. So you definitely don't want those like just floating around in the cytoplasm of the cell, damaging stuff that shouldn't be damaged. So that's a great example of compartmentalization right there, and why you need it. Central vacuoles found in plant cells. It takes up usually about 80% of the volume of a plant cell. But the purpose of that is because plant cells are usually bigger than animal cells. And by having that central vacuole taking up most of the volume, it pushes the organelles out towards the edge of the plant cell, which helps to keep homeostasis in check, because it's a shorter distance to actually transport nutrients and waste in and out of the cell. So, yeah. And that is it for concept 1. Sorry if I started talking fast towards the end, because the bell's about to ring and I did not want you to hear it. Anyway, guys, can't wait to see you in class, and make sure you watch this. I'll know if you didn't. Toodles.